considerations in the biotech industry and safety considerations that need to be considered throughout the process of scale up. We'll start by talking about the biotech industry in general. We'll go into some specifics about safety considerations, and then we'll go into details about different scale-up stages. So to give you a bit of background, I guess before we begin, I will say that we'll have a discussion at the end of the presentation. So if you have any questions throughout, feel free to drop them in the chat, and David is going to handle them at the end, or you can save them to the end and we can talk about them then. So a little bit of background about myself. I am originally from California. I did my undergraduate work at UC Santa Barbara, and then I came to Purdue Chemical Engineering for my graduate degree. My thesis work focused on material science, recombinant protein production, and using both synthetic and natural materials to create medical devices like surgical adhesives. Because of my research and my particular interest in the medical industry, I started working with Cook Biotech right after I graduated in 2018. And I've been really honored to be working there and working very closely on something so related to what I did my thesis on, which is quite nice. Cook Biotech is a medical device company. The focus of our products is on tissue regeneration, specifically to help tissue regrow where there is damaged or diseased tissue. And we generally tend to focus on making products with naturally sourced materials. So the photo you see here is of the stock material for most of our devices, which is called intestinal submucosa. It comes from pigs, and we clean it and then dry it and shape it into various forms for a wide variety of medical devices. So because of this field, most of my expertise at this point is in the area of medical devices. But a lot of the things that we're going to discuss today are relevant to the entire biotech industry. Specifically at biotech, what I do, I'm a development engineer. I work in the research and development department. But because we're such a small company, we only have about 100 full-time employees. I get to do a lot of different things. So in research, I look at new technologies and intellectual property filings. I evaluate new technologies from both internal and external sources, and I get to partner with academic groups to look at new studies and new technology. In the development side, I get to do product development, so taking those new technologies and trying to implement them into a product. So with that, I get to do initial testing on feasibility, and I get to contribute to submission packages to the FDA. And as kind of a hand in hand with the product development process is I get to do some process engineering as well. While we're working on product design, we end up sort of designing a process to make that product at the same time. And so throughout that process, we consult with our process engineering team, but we don't really formally hand off that process to them until the FDA submission occurs. So it's a pretty great role, I think, because I get to use both my science and my engineering knowledge, and I get to work on the front and the mid end of getting products to market. Now, when we're talking about biotech, I want to give a little bit of a definition first so we can kind of constrain the discussion. So how do we define the biotech industry? In general, biotech in industry is either going to be a product or a process that uses a biological material. So the food industry is a great example, anything with an animal or a plant source that's going into it. There is entire sections of the chemical industry that use biological inputs, either because they're digesting a plant to get to some chemical or use it as a feed source, or they're using microbes to create chemicals. And then there are also entire facets of industries, biologicals. These are things like cultured milk, where you are making a product that has bacteria or yeast somewhere along the line. On the other hand, you can also define biotech as a product or a process that makes something that goes into an organism, whether or not the product itself is from a natural source. So this is really where we're getting to high regulation areas with the FDA. This includes things like pharmaceuticals, medical products, and then biologicals, which are also things that have a biological source. So my background is in medical products. But today's presentation, we're going to focus mostly on that the medical industry, because a lot of the safety considerations are more intense than what you would see in food or chemical production, for example. One last thing to consider before we get into the weeds about safety is scale up versus process freeze. So as we are developing a new product and doing process design, there are either scale up steps, or at the end of scale up, we do process freeze. So scale up is any time where we're increasing the size of the process or the throughput of the process. And this can happen several times throughout the lifespan of the product. So generally we start out very small scale in the lab. We're working with micrograms, milligrams, milliliter scale products, usually like less than a liter at a time. It's something that one person can handle on their own. And then we eventually get to a pilot scale where we've moved it up and we're making prototypes. We need to produce more in order to support studies. And then we move to larger full scale production. And throughout these steps, there are a lot of opportunities to make changes to the process. But eventually we get to the point where what we call at Cook Biotech process freeze. And this is where we've settled on a process and we're freezing it in place. And that is the process that's going to be used for the package we submit to the FDA. And this is an important consideration because once you've submitted something to the FDA to be considered, any future changes to your process have a much higher level of rigor and inspection from the FDA. They're going to want to know, how does this process change that you're suggesting impact your product? But before that process freeze and before that submission happens, you don't have that intensity of rigorous oversight, so you have more flexibility. So usually that's the very last thing that's done. It happens often when you're starting to generate documentation for your process as well in whatever quality system you use. For Cook Biotech in particular, we generally have a relatively small scale. Because we're a small company, we're making specialty products. Even our full scale is relatively small compared to other giants in the industry. And so oftentimes we'll have our initial lab scale, and then our full scale ends up really being what might be pilot for another company. And we often do our scale up and our process freeze in parallel. And so those scale up considerations end up happening at the same time. So in general, in the biotech industry, there are sort of two categories of process safety considerations. In contrast to the chemical industry, oil and petroleum, if we have a safety incident, they tend to be less flashy. You're not usually going to get an explosion. You're not going to get a huge fume cloud that goes over the whole city and causes you to have to evacuate. But if there are incidents, they tend to be more silent and potentially more widespread. So say you have a batch of some product you're making and it becomes contaminated and you don't realize that it's become contaminated and it gets shipped out. You could potentially harm or injure hundreds of thousands of people and not really realize it until they start showing symptoms and you start getting in trouble with the FDA. So we sort of categorize these safety concerns into two areas: operator safety and end user safety. So in the case of operators, that is both the employees that are working on the floor at that full scale production, as well as the engineers who are interacting throughout scale up. And this category of safety falls under OSHA regulations. So in this case, we're concerned about physical harms and physical injuries, as well as in the biotech field, infection or exposure if we're working with a material that is can potentially infectious, like a bacteria or a virus, or if there are potentially bloodborne pathogens, especially if it's from a human source tissue. And these operator safety concerns are thought about throughout the process, because even in the lab scale, our operators are our scientists and engineers, and their safety is still critical. On the other hand, we have end user safety, and our end users are defined differently depending on the product. Sometimes our end users are like normal consumers for things that you buy on the shelf at the pharmacy. Sometimes the end user is a patient who's requested something that they're going to be interacting with at home, but it has to be prescribed. And sometimes our users are nurses and doctors, and the patient is not really the user because the nurse or the doctor is the one who's chosen the product and who is implanting that product into the patient. And so the safety of that end user is really critical. And the things that we consider there are contamination of the product. So is this product going to become adulterated in some way and potentially harm the end user? And the two main categories there are safety and efficacy. Safety is essentially, does this do something you don't want it to do that would harm the patient? And efficacy is, does it do what you say it's going to do? And when we have reduced safety, that's usually because something's become contaminated. And efficacy can be detrimental if it's either reduced or increased. So say you have a pharmaceutical and for some reason there's an incident in your process and your potency has increased twofold. That can have really negative effects on your patient population if it's suddenly super, super strong. In general, these sort of end user safety concerns happen around the process freeze stage. And this is because you want as much flexibility during scale up as possible as you're exploring your options for your supplies and your equipment and your different ideas about how the process is going to be designed. And that gives you more creative space to explore those options before you have to narrow in on your final selections near the end. So we're going to go into some more detail for both of these categories. We'll start off with operator safety. And in general, with operator safety, there's sort of three categories: chemical hazards, biological hazards, and physical hazards. And so chemical hazards, this is something I'm sure a lot of you are familiar with. If you work in a laboratory, it's exposure to hazardous chemicals. And there's a big difference as you're working towards scale up of the way that you interact with chemicals of different amounts, volumes, and masses. So if I'm working with a highly hazardous chemical, but I'm only measuring out like 40 mils and I'm pouring it into a beaker, and that's all happening in a fume hood, that's relatively easy to control and contain. But say that I have to do 40 liters of that. How am I going to actually do that pouring process? What's the risk of it splashing up onto me? Do I have any risk of it evaporating into the air? What are my ventilation controls? Does my user need to have any special PPE like a respirator? And so these are all things that we have to consider anytime we move up in scale. And especially for chemical hazards like this, we work hand in hand with our environmental health and safety team. And we look at regulations about volumes and masses of specific hazards and when we need to institute specific controls. And so other potential controls, like I said, specialized PPE. You can also have specific facility controls in the room itself, the way that things are venting, and then engineering controls, so additional isolators or additional equipment that are just designed to improve that safety. Beyond chemical hazards, we also have, if this is part of your process, a biological hazard. So like I mentioned before, sometimes you might be working with something that has a risk of harming your operator because of infection or exposure. And so in this case, usually what is done is you have, again, specialized PPE that helps to protect them. You also do specific training on how to handle those materials. But again, as you scale up, you're going to have more exposure to that and higher levels of risk. So you need to make sure that each time you change the scale, you check your assumptions and you make sure that the controls you have in place are still appropriate or potentially consider a change. For us, one big thing that we have to consider is disposal. So if you have just a small amount of some biohazard, it can be relatively easy to dispose of it in lab because you just dump bleach on it or you toss it in your autoclave. And then you can just dispose of it normally through the trash. But after you get to a certain level of biological hazards, you have to dispose of it in certain ways, and you may need to buy specialized equipment to do so, or hire out an external company to help you do that disposal process. And then finally, for operators, physical hazards are pretty important. It's not really something that is thought about often because we tend to focus on the technology and the process. But for our day-to-day operators, this is really, really critical because we want to ensure the longevity and the health of our employees. So these are things like repetitive motions. So I recently did a scale up on a process where we were filling syringes with a liquid solution to make a device. And in our lab scale, it was a 20 device batch. And so taking, a 50 ml tube of liquid and filling 20 syringes from it and just sitting on the bench, it's pretty easy. But when we moved to our scale up, we had it inside of a glove box. And so there's physical restrictions about how the operator interacts with the equipment. And we're increasing the size. And so now I'm making 200 devices. And so there's a risk of fatigue physically on that person's body. And there's also a risk of mental fatigue. And so there's an increased chance that they might make an error or mistake. Unfortunately, this device size is slightly too small to move on to something like automation. So when you're doing these sort of physical tasks, it's important to keep in mind that there are sometimes automation solutions once you get to a certain point where you can have a robot do it for you. Those tend to be expensive though. And so in this particular instance, we decided that this batch size was really too small to justify that expense. And we realized that our facilities really couldn't fit the equipment of the size that was needed. So we're moving forward at this stage with just continuing to use those human operators to do this device. But we try to build in process steps to reduce that fatigue on the operator. Another potential concern are bodily injuries, so things like sharps and slips. We have several processes in our facility that use a lot of water. And so we have to make sure we've included in our facility special mats and special shoes to reduce the risk of slips. That's not something that would really be a problem if I was just working alone in lab and I was working in a sink. But if I have a whole room of people with wash stations cleaning things, then that is a concern. And it's the same for sharps. A lot of our devices are produced manually. That means that we have someone sitting at a table and cutting and shaping material. And especially with those cutting equipment, there's a risk of laceration. And so once we've decided on our product configuration, we start doing the scale-up process. We try to engineer in steps to help protect that operator. So using specialized tools where the risk of cutting is lower by guiding the cutting tool or removing that cutting tool completely by using die punches or other kinds of equipment where you don't have an open blade. There's also the matter of physically moving things around. So if I have a stirred vessel with only a 1 liter reaction in it, it's pretty easy to just fill up the water or whatever and pour it in. But if I'm scaling up to a mid or a large size, that's not really possible anymore. And so at a certain breaking point, you start moving away from human use for lifting and carrying things, and you start moving over to equipment and engineered controls, pumps and piping and other things like that. And that threshold is different depending on the company, depending on the process, and depending on what the expectations are. So for us, some of our processes are set up to be done with pumping and piping systems, but a lot of our new processes and prototypes aren't, and we don't necessarily have the ability or the infrastructure to create that. And so we have to create safe ways for our operators to fill, carry, transfer, and pour these large volumes of liquid. So in general, across all these categories of operator safety, we have these sort of 3 scales that I mentioned before, are smaller, medium, and large. And the risks of each of those categories are different depending on the scale. So when we're starting at the small scale, we tend to have a few number of highly skilled operators, engineers, and scientists. They really know what they're doing, but sometimes they know what they're doing so well that they get sometimes a little too relaxed. And so you can have issues with your safety culture at that lab scale that need to be reinforced by training and reminders. In general, your volumes and your masses are small enough that you can just lift and carry by hand. You're using normal safety equipment is usually sufficient. You're doing a wide variety of tasks, but they're also less predictable because of that variety. So you often have to stop and check again, are my safety controls sufficient for this process I'm about to do? Are there any new dangers that I need to be aware of? But in general, if something does happen, the severity tends to be pretty low. So if you have an explosion, but it's in your fume hood, the risk of injury to a large number of people is relatively low. At the pilot scale, you're starting to involve more operators. They maybe don't have the same educational background, but they tend to be the most highly skilled operators at the facility. They've been there the longest. But you have to go through training processes with them to make sure that they understand what exactly is going on. Things are starting to get a little bit too heavy potentially, so you might have to start considering engineering controls, but they could be a little too small for that still. So you're sometimes in this awkward middle stage. You're starting also to need some specialized safety equipment, but depending on the budget for the pilot, you might not be able to afford that yet. So you have to sort of compromise sometimes. Oftentimes, lab scale equipment can be appropriate still at the pilot, but you'll need to buy new specialized equipment. The things that you're doing have gotten a little bit smaller in terms of variety. You're flexible in what you're doing, but you're still planning out the processes that are happening, so it's predictable. And because you have more people and larger equipment, your severity is increased a little bit. Then once you go to your industrial scale, you've got a lot of different people going on, working on this equipment, and the skill level and the turnover of those operators is really going to depend on the company and the company culture. So you may not be able to rely on having operators who really know what they're doing. At this stage, you're really beyond any sort of human scale to move stuff around. So you're designing equipment that is just doing what before could have been something simple, like moving water from one room to another or venting gas. And so you have all of this extra equipment that's being added onto your process to do these things like cleaning and venting, et cetera. You're having very highly specialized safety equipment that's just for your process, and your range of tasks is very narrow now because you're scheduled very strictly, usually at the facility level. But now your severity risk is very, very high as you're exposed to more people and you're using this huge equipment. So those are all of our concerns for operator safety. On the other hand, we're also going to consider user safety. So this, again, is something that is specific to the medical industry. It can also apply if you're making consumer products. But in general, in the medical industry, because of regulation, we're really considering these to a higher level and a lot more rigor. So we have, as I mentioned before, there's two categories, safety and efficacy. So the main concern for safety is, does the thing that you're making have any unintended side effects or any dangerous side effects? And this comes down to sort of two main categories, biocompatibility and sterility. So as we consider biocompatibility, this tends to focus not just on, so I'm leaving out product design in this lecture, because there's all of these things, especially in biocomp, that has to do with the product itself and what is in the product, the intended materials of the product. So we're not considering those right now. We're just talking about the process. So when I'm looking at my equipment and my supplies that are, especially those that are in contact with the product, whether it's a liquid or a solid or a gas, I need to be aware of the material that I'm using. So metals versus plastics versus elastomers, if they're in contact with the product, there's a concern potentially that they're going to leach into that product. And depending on the scale that you're working at, your choices may be limited. To give you an example, I recently designed a full-scale process that was too small to be considered by some design companies, some engineering design companies. We really were out of their scale. And so we had to look at off-the-shelf options. And usually when we're talking about metal, the metal of choice is stainless steel, and 316 grade steel is considered surgical grade. So we would prefer that, but the only off-shelf options was 304 stainless steel. So we had to consider the risk of using that grade of material and decide whether it was worth it cost-wise to use that material and accept those potential risks, try to mitigate them, or spend the extra money to do a custom 316 grade product. In addition, things like plastic have same sort of concerns. Sometimes you'll have a really specialized supply or part that you need that's not available in a pharmaceutical grade or a high quality, high purity grade plastic. And so you kind of just got to get what you can. And if the risk is high enough, again, consider contracting someone to make it custom from you from some stainless steel or something else. We also have to consider things about microbes. So in this case, we're talking about bioburden and we're talking about endotoxin. Bioburden is the amount of living things in our process, and endotoxin is essentially like the skeletons, the remainders of those living things. And we also think about those things when we're considering cleaning. So the material that we buy could sometimes have bioburden just from where we're getting it from. So we might have to clean it in advance, as well as cleaning in between processes. So we have to consider as we go through our scale up and we're increasing in our complexity and our size, how easy is it to clean or sanitize the equipment? And depending on the size of the equipment, we may be doing manual cleaning or we may be doing clean in place. And each of those processes have their own risks as well. So we need to be aware of that. If you can design a process where it is easier to clean the equipment, then that can be beneficial, especially when you're talking about narrow tubes or highly complex devices, or if you're working with solids that could cluster up and gunk up narrow gaps. If you have a really high risk process, or if you have a very hard to clean part, you may want to consider single-use plastics or single-use supplies, where you just use them once. They're usually shipped sterile from the vendor, and when you're done, you throw them away. So this, for example, we have a product we're making where we do a sterile filtration instead of a terminal sterilization. And because of that, we're making all of the contacting parts post-sterile filtration to be single-use. So we can be confident that once it is sterilized, it's only contacting sterile stuff, and then we package it and we ship it out. Now, along the lines of sterility, that's also an important thing to consider, especially if you're making medical products. So how are you going to sterilize the product? Now, if you're doing your lab scale work, usually sterility is not something you're thinking about yet, because you're doing mechanical testing where it doesn't need to be sterile, or you're doing a cell study or an animal study where you can sterilize it by hand very easily. And as you start to scale up, those may not be possible anymore. So to give you an idea, I recently worked on a product where we were partnered with one of our external companies, and they had been terminally sterilizing this product. And they found that it really negatively impacted the performance, because they were doing an autoclave and it was degrading the material. And so we had to work through with them to figure out, okay, we can't really do that. Maybe you use some other sterilization process in the lab, but now that we're doing this larger scale, we need to do a different sterilization method in order to not negatively impact this product. In general, if you're doing non-terminal sterilization, when I say terminal sterilization, I basically mean the sterilization is the last thing that happens before it gets packaged and then sent out to the customer. If you're not doing terminal sterilization, it's going to be higher risk, because your product is exposed to additional process steps after sterilization occurs. So you need to include more controls in your process and more testing steps to ensure that that sterility is maintained. On the other hand, we have efficacy. So efficacy tells us, does the thing that we're making do what we're claiming it's doing, and does it do it with high success rates? So if I'm making a product that is supposed to, cure X disease, but it only does it 10% of the time, we would not consider that to be efficacious. It could still be useful if there are no other therapies on the market, but that's how we consider efficacy. And as we're going through the scale-up process, we start to consider some specific logistical and design challenges around efficacy. So these are things like process limits and acceptable process limits. When you're running your process, you have different parameters that you control: temperature, pressure, stir rate, filtration grade, things like that. And you have to decide which of these process parameters and conditions could potentially impact my product quality. And if they do, what's my tolerance on the max and the min for that? At what point does increasing the temperature above a certain point negatively impact my product? And then you also consider variance. So you say, I want to set this process to 50 Celsius. Now I can set my controller to 50 Celsius, but in reality, it's not always 50 Celsius. It's above or it's below. So how much variability is there, and what's the spread of that variability, and how confident can I be that it's going to be that same way every single time? Sometimes it's important and sometimes it's not. So as an example, again, for sterile filtration, we're filtering a protein solution. We're heating the protein solution because it helps to increase the flow rate on the sterile filter. And we know that if we increase the temperature too much, it could potentially degrade the protein, which would impact the product quality. And so that is a very hard ceiling, and we try to keep those controls really tight. On the other hand, if the temperature is too low, it's just going to slow the filtration. It's not going to negatively impact the product at all. And so that's a less critical process limit. We still have a limit, because we know if it goes too low, then the filtration stops. But that's more of a process concern and less of a safety or efficacy concern. We also start to consider in our process verification and validation. So verification is when you measure something directly to ensure that it has a quality that you're looking for. So volume, if I pour some amount into a tube and I can see, oh, it's 50 mils, and I'm verifying visually that it's 50 mils. You can verify mass on a scale. You can verify temperature with a probe. Those are usually, if you can design a process for something to be verified, that's the better way to go. But sometimes you have a process where you can't measure it directly for whatever reason. Again, I'm talking about sterility a lot, but it's very important in the medical industry. Sterility is an example of something that cannot be verified because the process of doing the measurement for sterility is destructive to the product. You have to open it and expose it to a non-sterile environment in order to do that test. So in order to be sure that the process conditions are such that my sterility is maintained, I do what's called a validation. A validation is essentially a whole round of testing that you do that allows you to be confident that this process will create this condition, whatever, 100% of the time or an acceptable percentage of the time. And you rely on that because later the FDA is going to ask you, well, how do you know it's sterile? And you have to show them you did all of this testing to prove that your process is going to make a sterile product. Another potential example of validation is packaging. So when we package our devices, we put them into pouches that have to maintain that sterile barrier. And the ability of that barrier to stay closed depends on things like the pressure of the seal, the temperature of the seal, other factors like that. But we can't test that without destroying the package. So we validate the sealing conditions to be confident that if we operate this equipment at this temperature, at this pressure, it will create these conditions to create a sterile barrier. The downsides of validation is it gives you less flexibility as you go through your scale-up process. So you essentially validate a certain condition on your equipment, and you have to operate at those conditions and only those conditions. If you make any changes, you have to revalidate. But if you're verifying, you have a lot more flexibility. You can make a minor change and just say, well, I'm going to verify, I'm going to change the temperature on this thing, but I'm going to verify that the chemical reaction has occurred because I run this assay on every batch. So I don't have to worry about that. The last thing around efficacy is quality control throughout the scale-up process. So as we start to move things up, we want to consider, are we going to want to do this in a continuous or a batch process? Oftentimes in the medical industry, we default to batch processes because they're more convenient to do releases on and recalls on. Sometimes in addition, your input material may be inherently batch oriented. So for us, because we're using pig intestines, one sort of grouping that forms naturally is one pig worth of intestines. And so that way I can say, okay, all of these products here in this lot, whatever, were made from this particular pig at this particular farm. And that way I have that traceability. Doing a continuous process, depending on what you're doing, can improve your batch-to-batch homogeneity because you don't really have batches in the sense anymore. But if you have to do a recall, it's harder to decide where does one grouping end and where does one grouping begin. And as we're doing our quality control, as we're doing our testing for our release of our product, we have to decide things like what tests are we going to do? If we can't measure that thing directly, how are we going to test it? So if I'm making an adhesive, I'm maybe not going to be able to do all of these different adhesive tests every time. So maybe instead of doing actually adhesive testings on it, I'll say, as long as my product is this weight % of polymer and this volume in the tube, and I did this assay on the chemistry that I did that shows it has this many reactive groups, I'm confident that it will create this acceptable mechanical properties every time. And you also have to decide when you're doing that kind of testing, how many samples do you need to make, how many samples you need to test, and what kind of testing you're going to do. Again, destructive or non-destructive testing. So in conclusion, as we move through the scale-up process, we've identified that each step of the scale-up has unique risks. You'll want to check your assumptions and check your decisions every time to ensure that the choices you've made are still appropriate and still safe. You'll want to think about your employees and your operators throughout the entire process. You'll especially want to focus on user-related safety during that process freeze, if that's something that your company does. And you want to keep in mind that those early stages, the lab and the pilot scale, are really a very exciting and creative time that you should take advantage of and think very carefully. Because as you start to go through scale up, you generate momentum, mostly in documentation. You start to create paperwork around this process for the supplies you're going to use, and you start to build up all of these things that are related to your process. If you try to make a very strong process change later down the line, that's a lot of momentum that you have to push against. So try to be as broad thinking as you can very early on, and work through all your options before you start to go down, because once you start going down a pathway, you may not be able to leave that pathway. And I'm happy to take any questions, as well as if you'd like to contact me at another time, my email is here, and our company website if you're interested in learning more about the kind of products that we make.
Thank you so much for the fantastic talk, Sydney. Does anyone else have any other questions? I had a couple, but I'll let other people start.
Hey, Sydney, I have a question. So I'm wondering, in your experience, or in your opinion, if you haven't experienced this yet, what is the biggest contribution to possible recalls or contamination in biomedical products? Like, where along the line does that happen?
That's a really tough question because it really depends on the company and the product and what they're doing. I will say probably the most common in terms of numbers reason for recalls is like mislabeling. Like, it's not necessarily even something that affects the safety or the efficacy at all. It's just that some label was put on wrong or was put on the wrong package. And that's enough to trigger a recall because your end user may not realize that and use the product incorrectly. So in my understanding, that's the most common. There are actually publicly available databases for recalls by the FDA. So if that's something you're interested in learning about, it's really easy to look up. They explain the reasoning very clearly on all of the recalls. And a lot of them are low risk, like seemingly just like mix-ups by the employees.
That's really helpful. I kind of have a follow-up question. So if a lot of a lot of these possible recalls tend to be like lower risk issues, is it like so you're like talking about in a small company, you're a big part of who assesses where the safety risks are and how to mitigate those. So is there like, is this like a conversation you have with your boss? Is this a conversation you're having with the FDA? Like, who ensures that you're addressing all possible safety concerns?
That's a great question. So what you're asking is who has that oversight at what stage?
Yes.
So there is lots of oversight at multiple stages. So pre-submission to the FDA as we're developing the process, and every company operates differently. But for us, in the early feasibility, it's usually limited to that team if they consult with outside groups potentially. But once we start creating documentation, there are formal processes that we go through where the process is reviewed by a dedicated team whose job is to review process safety. And they're represented by a variety of departments. So we have process engineering, we have quality engineering, we have regulatory, all these different people in this group to help us ensure that the process is well designed. Then when we go to FDA submission, we basically have to justify our choices, and the FDA will also question and prod us to ensure that what we've done is correct. I will say that the FDA, they don't really care how you make something necessarily. All they care is that the product is safe and effective. So whatever you've done, the details matter, but the way you've done it doesn't really matter. Kind of like, you know what I mean? Yeah.
Great, thank you.
I have a question still regarding these recalls. What happens to them? Do they come back to the company and maybe the company is looking into what was wrong, try to sort out the problem and reuse them, or they are just, out of waste, let's say?
That's a great question too. So the whole process of interacting with the FDA in general is very formalized. So in the case where you have some product that's been released and there's a complaint, so there's a whole process of filing complaints. And if enough every complaint has to be reviewed by the company and by the FDA. And if enough complaints have accrued for a particular issue, then they'll review it to potentially trigger a recall. If they've triggered a recall, they inform the company. The company has a set amount of time to respond, have a discussion with the FDA. They work together to do a problem discovery process to figure out what potentially caused this issue. They'll decide together what needs to be done, whether the product needs to be destroyed or whether it just needs to be stopped being sold, or if it can be recalled and then fixed and then sent back out again. But it's very like regimented, and if you really have to stay on top of it, because if you fall behind for whatever reason on the company side, you can get into some trouble because the FDA might think that you're trying to be not responsible in your production of your product.
Well, thank you for that. I also have a question about the first part, what you talked about in the presentation. How long is the process of preparation for the animal tissue to be defined as good enough to be used for using medical devices for people? So
are you asking?
I guess it takes a few stages, and then you have to verify or validate that yes, this is at a good standard for it to be used for human medical devices. But maybe you can answer that.
Yeah. So the process of proving that any material is safe to put into a human is really the same regardless of what the source material is, whether it's animal tissue or plastic or metal. And that's a multi-stage process. Usually you have to get some levels of approval from the FDA before you can start putting it into humans. So you have to show that it's safe, and usually you start off by doing cell studies to show that the cells don't respond negatively. Then you most of the time do an animal study. Sometimes the animal study is skipped depending on how well established that material is. If you're using a material that's well known to be safe, then you don't usually have to show as much evidence or proof that it would be safe. But yeah, even for our pig intestine or any other material, there are usually specific tests that the FDA requires to show before they will allow you to do your first in human trials.
Thank you.
I had a question about operations. So, like, what would you say is the biggest hazard to operators working on like a large scale process in the medical device industry? I'm just curious.
The biggest hazard to operators. It depends a lot on how your process, how much human intervention is in your process versus automated. So there are entire facets of the medical device industry, especially in traditional devices, that really operate more like a factory. They have a lot of automated equipment doing the machining and assembly of the devices. And so in that instance, the hazards are the same as in any factory for operators, risk of crush, risk of exposure to those kinds of equipment. If you're talking about, like for us at Cook Biotech, we have very little automation. And so really the majority of our operators, and that's why I mentioned it, the highest risk, the highest frequency risk is like lacerations, because they're handling this material, they're handling sharp devices to cut and manipulate the intestine. And so that really is the highest risk there. If you're talking about other areas of biotech in like pharmaceutical or antibiotics, where they're working with equipment to do fermentations or things like that, Then the risks are similar to the chemical industry, where you're concerned about pressurized equipment or high temperature equipment. Usually the temperatures aren't quite as high as in the chemical industry. Large volumes of liquids, things like that.
Okay, yeah, thank you very much.
I have another question. So I'm really intrigued by the idea that Cook Biotech actually doesn't use much automation. I always imagined any product company using like 100% automated everything. And so I'm wondering, like, at what scale, like not abstractly, but like concretely, how many products do you need before you really start to consider automation in a process?
Yeah, that's a great question. So there's sort of two parts to that. I'll start off by saying the motivation to include automation is really company culture dependent. So Cook Biotech, as a subsidiary of Cook Medical, has a big emphasis on choosing human operators over automation. It's part of our company culture. It's part of our legacy. And so in general, when we think about processes, we tend to emphasize processes that require a lot of human dexterity that, an equipment machine may not really be able to do. In terms of scale, it's sort of a complex decision, and it's not just a technology decision, it's also a budget decision. And so usually what you have to do is look at your yields and your throughput at the two different processes. You look at labor time. So in general, labor time is the most expensive part of any process. So the number of operators and how long it takes them to make it. You look at the cost of the automation equipment and the return on your investment. So how long will it take for this $10,000 piece of equipment to, return us a profit on this particular process? And often for a new product line, you're going to be, especially in the medical industry, it's going to be years before you're producing it at any scale that you might get that return, because the first several years, you're only producing enough product to support a clinical trial, which is going to be tiny. And then you also have to look at your disease state you're targeting. Your market might be so small that doing automation is not really necessary, because you're only going to make, one batch of 200 devices twice a year or something like that. And it's not worth it to make an equipment that can create 10,000 devices a month.
That makes sense. Does that mean that like automation is considered often after the process freeze stage, like after it's been approved, after you've kind of made you've done your initial tests, preclinical tests and trials and whatnot, and then people find it's a good product and then consider automation, or it cannot? You're shaking your head.
No, because once you submit to the FDA, your process is stuck. And so even if your product, so there's a very complex market analysis that goes on when you even choose a specific product line to focus on for development. And part of that is an analysis of how many patients can you access. And so you'll know in advance, or you at least have an estimate of, once we get to approval and once we start selling, in the next 5 years, in the next 10 years, how many devices do we think we're actually going to be making and selling? So we've already anticipated that, because once we get to that approval with the FDA, any change to the process is very difficult to do, especially something that big like adding in that automation, because now there's a whole new round of validations that have to be done on that equipment. So the likelihood of that happening is usually pretty low.
Any other questions? Well, if not, we'd like to thank Sydney very much for joining us here today. This was a wonderful talk. I certainly learned a lot. I know nothing about the medical device industry, so this was very informative. So yeah, thank you very much.
Absolutely. It was my pleasure. Thanks for inviting me.
I'll go ahead and stop the recording.